Good morning, everybody, from a snowy, very cold Washington, D.C. area, although I think we fared a lot better than some of our colleagues to the north in the storm. Well, I am very excited for the opportunity to address you all today. I'm personally a big fan of the mission of CDER's SBIA group and so happy that I can be here to discuss some current activities and recommendations on behalf of my group, the Division of Microbiology Assessment. I'm a microbiologist and acting quality assessment lead in DMA, and I also presented in this session that was held last summer, where I spoke on recommendations for the control of non-sterile products. Today, I'll be talking about some of that material, along with taking a deeper look into some aspects of our recommendations and providing some updates. In addition, since we have multiple DMA speakers today, and I'm the first, I'll be giving you a quick overview of our division's organization and activities. In 2015, many of the groups involved in assessing the quality aspects of a drug product, such as chemistry, manufacturing, and microbiology, were organized into one FDA office, and that's the Office of Pharmaceutical Quality. The purpose of this organizational scheme was to serve as a single voice from CDER that assesses and communicates quality standards for drug products throughout their life cycle. OPQ consists of a number of offices which oversee the various aspects of quality, all the way from clinical trials to the marketing applications for generic products. The organization allows for better communication between and among the quality groups, and DMA is part of the Office of Process and Facilities within OPQ. DMA is placed in that office chiefly because of the large role that processing and environmental conditions have in the manufacture of a product that's of good microbiological quality. DMA's mission statement, which I've listed here at the bottom of the slide, reflects the nature of the very diverse work we do, the different types of applications we work with, the variety of products we work with, the various subject matter expert roles we play in CDER and the agency. Now, if you think you've seen this slide before in another presentation, but that it's grown considerably since the last time you saw it, that's not your imagination. DMA has grown tremendously since its creation as part of the OPQ reorganization, and we're still growing. Quality microbiology has been recognized as a critical aspect of the manufacturing process, and to address increasing demands on our time and expertise, staffing has increased accordingly. Many of you have likely interacted with a member of our group as we work very hard to participate in outreach activities. You may have heard a talk from our director, Len Enzer, one of our 4 branch chiefs, or one of the quality assessment leads who comprise our DMA leadership team. Something to keep in mind if you interact with or hear from DMA, and that's that branches 1 through 3 of the group are involved with review and activities associated with small molecule drugs. These branches are involved in the review at all stages of development, from IND applications through new and generic drug marketing applications. This presence in the development process allows us to provide clear, consistent feedback to direct sponsors and applicants through the regulatory process as it concerns microbiology. Branch 4, which is involved in the review of biotech-derived large molecule products, is also involved in all phases of the development process. In addition to these review activities, Branch 4 members play a large role in the inspection of facilities involved in the manufacture of both drug substances and drug products. In addition to the day-to-day work of review and inspection that take up most of our time, DMA plays a larger role in the agency by serving as subject matter experts for a variety of CDER and agency activities. DMA members are involved in policy development, whether that's internal guidance and regulation writing, or working with external organizations to write technical standards. And finally, DMA is very involved in outreach activities such as today's presentation, and we're also able to take advantage of a number of training opportunities, such as those available through the Parenteral Drug Association, to help us train our large numbers of new staff. All right, so now that I've given you some insight into the organization of DMA, and since we're going to be hearing from some other DMA speakers today, you can kind of know where we're coming from. Let's switch over to the technical part of my material. My topic focuses on our current recommendations and considerations for the microbiological control of non-sterile products. We'll be expanding on some of the areas that I discussed when I presented for the SBIA group last summer. In CDER, and certainly in DMA, we're always asked to consider the risk to patients that particular drug quality attributes are intended to address. When considering non-sterile products, the major risks are patient exposure to excessive numbers of microorganisms and exposure to pathogenic microorganisms. There's also the additional risk that too much microbiological activity in a product can degrade it chemically so that it no longer has its intended efficacy. The intent to guard against these risks is behind every recommendation, every item that we in DMA consider when we're assessing non-sterile products. There's a tremendous diversity in non-sterile products that comes first from their various routes of administration. This makes the establishment of a patient risk profile for these products a daunting prospect. Additionally, some products are intended for patient populations that may not have the robust immune function that protects them from exposure to microorganisms. Finally, there's a tremendous diversity in the types of formulations seen for non-sterile products. With all these considerations, there's really no one-size-fits-all risk mitigation strategy. So how can we begin to approach the idea of risk assessment for non-sterile products? Well, it comes down to knowing the source of any risks and controlling those aspects accordingly. For non-sterile products, risks come from either situations where the product is contaminated, particularly with objectionable organisms, or the microorganisms that normally exist in a non-sterile product are allowed to proliferate to undesirable levels. The steps that prevent these occurrences are an appreciation for the roles that product formulation and manufacturing control play. Since there's no one-size-fits-all strategy, these items are critical to consider on a product-by-product basis during development. Now, in the next few slides, I want to talk about one of the biggest risk factors for the quality of non-sterile dosage forms, and that's the water that's present in a formulation. You can see my Mars rover picture there. And why did I put that in a microbiology talk? Well, there's a reason why so much effort and attention is given to the search for water on other planets. And that's because all life as we know it depends on the presence of water. Water is required for microbial proliferation, and in any circumstance where there's water, there's a risk for that proliferation. Since non-sterile products naturally contain microorganisms, products that contain water are at risk for proliferation if there are no appropriate mitigating factors in place. Now, notably, there are some non-sterile products that don't contain any water, and they're a lower risk. This lower risk is reflected in testing requirements, such as USP 51's requirement for the demonstration of antimicrobial effectiveness only in aqueous dosage forms. And one additional point to make, and this might sound a little silly, but you'd be surprised by how frequently it comes up, is that not all aqueous products are liquid, and not all liquid products are aqueous. There are many semi-solid dosage forms that contain water, and they're therefore at risk, while there are some liquid dosage forms that contain no water at all. Alright, so I would like to get some feedback from you guys this morning. So I am interested in seeing how many of you out there are familiar with the concept of water activity. Alright, so we're coming in looking like people have heard of it, but they'd like to learn more. That's a good place to be. Not a lot of experts out there. That's not surprisingly. It's not a very widely discussed concept, but I think it's something useful that we can think about for non-sterile products. All right, so coming in, bringing in the results. And all right, well, I think when I saw it last, most of the people had heard of it but wanted to learn more. So let's talk a bit about water activity, the concept and some practical applications. All right, there we go. Let's talk a bit about water activity. So this is a concept that's actually been recognized in the food industry for decades as a risk factor for the microbiological spoilage of foods. Physically, water activity is a measure of the partial vapor pressure of water in a substance as compared to the vapor pressure of pure water, and that's what the equation on the slide reflects. The highest water activity, that of pure water, is 1, and other water activities are expressed as a decimal value between 0 and 1. Now, for a more practical, and the true experts would say overly simplistic definition, but we'll go with it, water activity is a measurement of how tightly associated water molecules are with other molecules in a substance, whether or not the water is bound to those molecules. Well, how does that relate to microbiological activity? Well, think of fresh fruit versus fruit that's been turned into jam. When making jam, you add lots of sugar, which binds with the water that's present in the fruit. The sugar binds to the water, lowering the water activity, and the bound water is no longer free for microorganisms to use for proliferation, which prevents the spoilage of the jam. Now, it's important to note that water activity is a separate concept from water content. In fact, two different substances may have the same water content, but a different water activity. Generally, products with a water activity of below 0.6 are low enough that microbial proliferation is not possible. So these products are very low risk. Being such low risk often qualifies these products for reduced microbiological testing schedules, which we'll discuss a bit towards the end of the presentation. On the other hand, many products have a high water activity and must seek other ways to limit microbial proliferation. The chart here at the bottom of the page, or bottom of the slide, is one from USP's very good chapter 1112 on water activity. I've copied a portion that shows some information on products that have high water activities, common contaminants seen in these products, and USP's testing recommendations. There are additional examples in the chapter, and I encourage you to check it out as a resource. Now, knowing the importance of water and water activity from a microbiological perspective allows us to divide very diverse non-sterile dosage forms into three very broad categories. First, we have the solid dosage forms, such as tablets, which are in their own group because of their low water activity. Since solid dosage forms typically have that low water activity, they're low risk for microbial proliferation over shelf life. Next, we have the non-solid dosage forms, which include both aqueous and non-aqueous products. Non-aqueous dosage forms are distinguished because they either have low water activity or they just don't contain any water, and a very low risk for proliferation. Aqueous products, those containing water with an activity of greater than 0.6, are the riskiest dosage forms for proliferation and must have additional formulation considerations in place for stability over shelf life, such as the inclusion of antimicrobial preservatives. So, we've been talking about how product formulation can contribute to the risk of microbial proliferation. Now let's talk a bit about the role the manufacturing process plays in introducing risks and talk a little bit about how they can be mitigated. When considering the manufacturing process, the risk of product contamination starts to come into play. Sources of contamination include the components that make up the product. Water systems can be significant sources of contamination, which we'll have some real-life examples of later in the presentation. Care should be taken to ensure that water systems are appropriately maintained and monitored. The manufacturing environment can put product at risk, particularly the presence of operators. Now, I know we don't like to think of ourselves just as big walking bags of microorganisms, but the truth is that operators are a significant source of microbial contamination in any process. Finally, the risk of proliferation is not absent in the manufacturing process. Lack of control and process holding times can allow for proliferation, which is then passed on to the finished product. Now, any well-designed and controlled manufacturing process will culminate in finished product testing. The most widely applied methods for non-sterile products are the test for total microbial enumeration, as described in USP 61, and the test for the absence of specified microorganisms, as described in USP 62. However, unlike the other compendial microbiological tests, these chapters don't contain any acceptance criteria. And this is where the recommended acceptance criteria from Chapter 1111 come into play. And these are listed on the right hand of the screen. They may display a little small, but they're just there for illustrative purposes. One interesting point about the acceptance criteria in 1111 is that they're grouped by route of administration. They recognize that different routes of administration can tolerate the introduction of varying microbial loads, and that different body sites are at risk from different organisms. For example, E. coli should be absent in oral preparations, while cutaneous preparations focus on the absence of Staph aureus and Pseudomonas aeruginosa. In some circumstances, reduced microbiological testing can be appropriate for very low-risk products, including those with low water activity. However, do note that for this type of strategy to be successfully implemented, much thought must be dedicated to upstream controls. This type of strategy is typically reserved for solid oral dosage forms because the risk profile from both a perspective of formulation and from the route of administration, an oral route of administration, are very low. Another area of consideration for product testing is antimicrobial effectiveness, or AET. As you know, if a product is aqueous and multi-dose, it has to either contain a preservative or demonstrate that it's self-preserving, typically by testing according to methods described in USP 51. During product development, it's important to perform AET on product that contains preservative that's at or below the lowest specified amount, which is usually around 80% of the label claim. And after that developmental testing is performed, preservative content may be used as a surrogate for some testing time points. However, I do want to emphasize one item which we've been seeing issues with in applications, which is that regardless of whether preservative content is used as a surrogate for testing, AET should be performed on at least one batch at the end of the product's shelf life. This is in agreement with recommendations in ICH Q1A and is important to demonstrating that the preservative has the proper biological activity over the shelf life of the product. We've dealt with some incidents recently in which products pass preservative content testing over time, but fail AET. So testing over the product's lifespan is something that we're trying to emphasize more heavily when we see these in applications. One final issue that we deal with frequently in applications is in regards to testing for the members of the Burkholderia cepacia complex, or I'll be referring to them as BCC. These organisms are a particular issue for aqueous products, as they're frequently associated with aquatic environments, or more appropriate for our considerations, facility water systems. BCC are capable of forming biofilms, which can make it difficult to eliminate them from the environment once they're established. BCC can survive and proliferate even in the presence of preservatives or other antimicrobial agents, and can utilize a variety of carbon compounds for energy that other microorganisms are not capable of. BCC can be difficult to culture, as they don't tend to grow well when they're shifted from a low-nutrient environment, like a manufacturing facility or a drug product, to a rich one, like microbiological media. So care must be taken when developing sampling methods. BCC have been implicated in a number of public health events and product recalls involving both sterile and non-sterile products. DMA is now routinely requesting that applicants pursuing approval for non-sterile aqueous products establish a BCC risk mitigation strategy for the manufacturing process, as well as an in-product testing strategy to demonstrate the absence of this microorganism in the product. It's important to note that testing for this microorganism is not currently outlined in USP's Chapter 62, and without an established compendial method, validation studies must be performed, so we're working hard to get the word out on this so that applicants aren't surprised during their review cycle. Although it's my understanding that USP is planning to undertake development of a method for the detection of BCC in the near future. All right, Another opportunity for me to get feedback from the audience. So, if your company manufactures non-sterile products, has a risk assessment for BCC contamination been performed? It's like people aren't sure. We do have some people who have performed a risk assessment, and they do also conduct routine monitoring activities, which I think is kind of the gold standard for this. But looks like most people haven't performed any BCC risk assessment. Okay. All right, so let's go back to the slides. I am not a big fan of trying to scare people into doing things. I don't think that's an effective way to deal with people. But the next couple of slides I have do discuss some really important public health events that we have dealt with over the last year with Burkholderia cepacia. And I just wanted to share those with you just to give you an idea of how widespread this issue is within the pharmaceutical manufacturing community. One fairly high-profile event involved the contamination of a stool softener product and affected a number of vulnerable patients in 8 states, leading to illness and death for some. The source of the Burkholderia was finally determined to be the water used to manufacture the product. So again, back to importance of monitoring water systems for this organism. Ultimately, this product was voluntarily recalled by the company. Burkholderia cepacia contamination also led to a Class 1 recall of filled flush syringes. This one might be slightly out of place in this presentation as it deals with a sterile product, but this was a significant event nonetheless, with over 100 infections in 5 states. CDC reports that there were 7 deaths among the cases. However, since the affected patients were ill and hospitalized at the time of their deaths, they can't be contributed conclusively to the Burkholderia infections. A voluntary recall was also initiated for chlorhexidine wipes. Fortunately, there were no adverse events associated with this. I'm not aware if there was a conclusive determination of how the Burkholderia was introduced into this product in the manufacturing facility, but do note that the contamination was detected in the presence of chlorhexidine gluconate, which is a pre-surgical skin prep disinfectant, very harsh antimicrobial. This is a great example of how Burkholderia can persist in the presence of antimicrobial compounds. Now, I won't speak too much regarding our involvement in an incident involving Burkholderia multivorans in a non-sterile nasal spray, since my colleague John Metcalf is going to do an entire presentation on it next. John's presentation is going to go into details about CDER's working process for dealing with adverse event reports, and it's quite an interesting story. I'm glad he's able to give it to you today. And finally, while Burkholderia contamination events have been the bulk of activity over the past year, other contaminants are also a significant concern, including incidents involving Pseudomonas contamination in a topical product and Staph aureus contamination in shampoos and lotions. All right, we've covered so much in this presentation, and I'd like to leave you with some parting thoughts on the subject. So first off, everything that we do in DMA, and truly everything in CDER, is focused on mitigating risks that drug products present to patients. In this case, we're trying to prevent issues stemming from patients being exposed to too many or the wrong kinds of microorganisms. And events like this occur if products are either contaminated or if microorganisms are allowed to proliferate uncontrolled in the product. Steps that manufacturers can take to mitigate that risk include considering the product's formulation and its manufacturing process. There's no one-size-fits-all approach to risk mitigation, so it's something that has to be considered with knowledge of the process and the product in mind. Finally, proper endpoint testing is critical to demonstrating that the controls are functioning and are in their proper place. When I spoke on this topic last summer, I mentioned that DMA was in the process of publishing our recommendations on considerations for non-sterile products. At the time, we were optimistic that these would be published in 2016, but as you can see on your calendar, 2016 has come and gone with no such publication, and nobody is more disappointed about this than I am. The document that DMA microbiologists have produced outlining our recommendations is still in the clearance process. However, we're hopeful that it will be published soon and will provide you with insight to our concerns, recommendations, and expectations. Here's my contact information. I will be presenting in the upcoming Q&A session, but would also be glad to communicate directly. And finally, on behalf of all of us at DMA, we appreciate you taking the time to interact with us today, and I thank you so much for your kind attention to my presentation.